All right, let's dive into a clinical mystery that you'll see time and time again, both in the hospital and, you guessed it, on your board exams. We're talking about a sudden, really dangerous drop in red blood cells, and we're going to start with a classic case that every clinician has to know. So picture this. A young guy, perfectly healthy otherwise, comes in, he's just feeling weak, kind of wiped out, and his skin and eyes have this yellowish tint. He's jaundiced. The story is, he just started an antibiotic a few days ago for an infection. You get his labs back, and whoa, his hemoglobin has absolutely plummeted. He's in the middle of an acute anemic crisis. So you gotta ask, what on earth is going on inside his red blood cells that's causing them to just self-destruct like this? Well, the answer lies in a hidden genetic vulnerability. Okay, so our investigation starts by putting a name to the culprit behind this whole situation. The condition we're talking about is glucose 6-phosphate dehydrogenase deficiency, or let's just call it G6PD deficiency from now on. So what is it, really? Well, it's a hereditary enzyme defect. This isn't something you catch, you're born with it. And here's the thing, it doesn't cause problems all the time. It only rears its head in these specific episodes of what we call hemolytic anemia. Hemo means blood, and lytic means to break apart. So his red blood cells are literally bursting. And the trigger for this is always the same thing, oxidative stress. Now, who gets this? This is super important. G6PD deficiency is a classic X-linked recessive disorder. That is a huge buzzword for your exams. Since men only have one X chromosome, if they get the faulty gene, they get the disease. That's why it primarily affects men. And it's really common in people of African, Mediterranean, and Asian descent. Get this, it affects a staggering 10 to 15% of American Black men. Okay, so we know what it is. But to really get it, we need to understand why it happens. We've got to go inside the red blood cell and look at its most important defense mechanism, its oxidative stress shield. The best way to think about this is with a simple analogy. The G6PD enzyme, it's like a shield. Its whole job is to protect the red blood cell from damage. And the things attacking it, these fiery arrows, are oxidants. You know, things like infections or certain drugs that create a really stressful environment inside your body. So check this out. In a normal cell, that G6PD shield is tough. It can handle an oxidant drug, no problem. The red blood cell stays protected. But in a G6PD deficient cell, that shield is weak. So the oxidative stress just punches right through, causing the hemoglobin inside to warp and clump together. These little clumps are called Heinz bodies, another key term, and they wreck the cell from the inside out, basically marking it for destruction. All right, so we know the mechanism. Now, how do we prove it in a patient? Well, like with so many blood disorders, the clues are hiding in plain sight, right there in the patient's blood. It's pretty amazing, actually. When we take a single drop of blood, smear it on a glass slide, and look at it under a microscope, what we call a peripheral blood smear, the whole story of what's happening just becomes crystal clear. And the first thing you might see is something called a bite cell. And no, that's not a metaphor. It literally looks like someone took a bite out of the side of the red blood cell. So what's happening here? Well, as these cells pass through the spleen, special cells called macrophages see those Heinz bodies we just talked about, and they pluck them out, taking a chunk of the cell membrane with them. Wild, right? The second big clue is the Heinz body itself, that clump of damaged hemoglobin. But, and this is a huge clinical pearl, a classic exam question, you are not going to see Heinz bodies on a standard blood smear. You need a special supravital stain, something like crystal violet, to make them show up. That's a point you absolutely cannot forget. Okay, this brings us to a really, really important clinical decision, a real-world challenge. Your patient is clearly breaking down red blood cells. You suspect G6PD deficiency. So do you order the test for the enzyme level right now, in the middle of the crisis? The answer is a hard no. Absolutely not. You have to wait. The only right time to test is weeks after the acute episode is over. Testing too early is one of the biggest diagnostic mistakes you can make. So why? What's the logic here? Well, think about it. During an attack, it's the oldest red blood cells, the ones most efficient in G6PD, that get destroyed first. So the body panics and starts pumping out a flood of brand new baby red blood cells, which we call reticulocytes. These fresh cells straight from the bone marrow have normal G6PD levels. So if you test during the crisis, you're mostly measuring these new cells, and you can get a completely misleading, falsely normal result. All right, we know how to diagnose it, so now let's focus on what causes these episodes to begin with. What are those fiery arrows we need to be on the lookout for? The most common trigger, hands down, is infection. But for your exams, you absolutely have to know the drugs. The high-yield ones to burn into your brain are dapsone, the antibiotic nitrofurantoin, the antimalarial primaquine, and especially sulfa drugs like Bactrim. Quinolones are also on the list. And of course, there's that classic trigger you hear about for the Mediterranean variants, fava beans. Okay, finally, let's bring it all home with a management plan. What do we actually do for a patient once we've diagnosed them with G6PD deficiency? Well, the really good news is that the treatment is incredibly straightforward. The cornerstone of management isn't some complex drug regimen. It all comes down to one word: prevention. So the strategy is pretty simple. First, you educate the patient. They need to know what triggers to avoid. Second, if an episode does happen, you just find and stop the offending agent. And third, and this is key, the hemolysis is self-limiting. Once that trigger is gone, the body takes care of the rest, clearing out the damaged cells and making new healthy ones. Okay, let's tie this all together with the ultimate high-yield buzzword summary for G6PD deficiency. If you remember nothing else, remember this. Inheritance, X-linked recessive. Pathophysiology, oxidative stress leads to hemolysis. Smear findings, you're looking for bite cells in Heinz bodies. Key triggers, infections and sulfur drugs. And management, simple. Avoid oxidant triggers. That's your cheat sheet right there. And that leaves us with one last, really fascinating thought. If this enzyme deficiency can make people so sick, why is it so incredibly common in certain parts of the world? Why hasn't evolution just gotten rid of it? Well, the leading theory is that it might actually offer some protection against malaria. It's a powerful reminder that what we see as a disease in one context might just be a life-saving advantage in another. Pretty cool, huh?